Biomarkers in Rare Diseases, Challenges and Opportunities. Title: Biomarkers in Rare Diseases, Challenges and Opportunities. Introduction. Rare diseases, often referred to as orphan diseases, affect a small percentage of the global population. Despite their individual rarity, rare diseases as a group impact the lives of millions worldwide. Biomarkers, which are measurable biological indicators, play a crucial role in the diagnosis, understanding, and management of rare diseases. This article explores the challenges faced in identifying biomarkers for rare diseases while emphasizing the opportunities they present in advancing diagnosis, treatment, and research. Understanding biomarkers in rare diseases. Biomarkers, broadly classified as genetic, molecular, or cellular, are measurable indicators of biological processes, diseases, or responses to treatments. In rare diseases, these markers hold significant potential for early diagnosis, monitoring disease progression, measuring treatment effectiveness, and identifying suitable therapeutic targets. Biomarkers serve as invaluable tools in bridging the gap between fundamental research and clinical practice, offering hope for patients with limited treatment options. Due to the scarcity of patients with specific rare diseases, it becomes arduous to collect statistically significant data, hampering biomarker discovery efforts. 2. Heterogeneity of rare diseases. Rare diseases encompass a vast array of conditions, each with unique genetic, molecular, and clinical characteristics. The inherent heterogeneity within these diseases poses challenges in identifying unified biomarkers that can be universally applicable. Moreover, biomarkers can differ not only between diseases but also among patients with the same rare condition. 3. Lack of validation. Developing reliable biomarkers requires robust validation through independent and rigorous studies. However, the rarity of these diseases often limits access to sufficient samples for validation studies. This scarcity of data provides an impediment to building confidence in the accuracy and reliability of potential biomarkers. 4. Variability in disease presentation. Rare diseases often exhibit a wide spectrum of clinical manifestations, making diagnosis challenging. The variability in disease presentation further hinders the identification of consistent biomarkers. It is crucial to recognize these variations to ensure biomarkers accurately represent the underlying disease processes. Early identification can lead to timely interventions that may enhance patient outcomes. Additionally, biomarkers can facilitate prognostic assessments, providing information on disease progression and enabling personalized treatment plans. This approach holds promise for more efficient and targeted clinical trials, resulting in better evaluation of potential therapies and optimized treatment approaches for rare diseases. 3. Tracking treatment response. Biomarkers provide a means to objectively assess treatment response, enabling more efficient monitoring of patients during therapeutic interventions. By tracking changes in biomarker levels, clinicians can gauge the effectiveness of treatments, adjust dosages, or switch to alternative therapies when necessary. 4. Accelerating drug discovery. Biomarkers offer opportunities to identify novel therapeutic targets by revealing underlying disease pathways. Advances in biomarker research for rare diseases. In recent years, there have been significant advancements in biomarker research for rare diseases. These developments have the potential to address some of the aforementioned challenges and provide new opportunities for diagnosis, treatment, and research. Some of these advances include 1. High-throughput technologies. The advent of high-throughput technologies, such as next-generation sequencing and proteomics, has revolutionized biomarker research. These technologies allow for the analysis of large numbers of samples simultaneously, enabling researchers to identify potential biomarkers more efficiently and accurately. 2. Collaborative research networks. The establishment of collaborative research networks, both at national and international levels, has facilitated the sharing of resources, data, and expertise. These networks bring together researchers, clinicians, and patient communities, allowing for larger patient cohorts and more robust validation studies. This collaboration is essential in overcoming the limitations imposed by the rarity of these diseases. By aggregating and analyzing diverse datasets, researchers can uncover patterns and correlations that may not be apparent in individual studies. This approach improves the statistical power and reliability of biomarker discovery efforts. 4. Artificial intelligence and machine learning. Artificial intelligence and machine learning algorithms have shown great promise in biomarker research. These techniques can analyze complex datasets and identify patterns that may be missed by traditional statistical approaches. By leveraging these technologies, researchers can enhance the accuracy and predictive power of biomarkers, leading to improved diagnostic and therapeutic strategies. Looking ahead. Despite the progress made in biomarker research for rare diseases, there are still numerous challenges that need to be addressed. The limited patient population, disease heterogeneity, and lack of validation continue to pose obstacles. However, with continued dedication and collaboration, these challenges can be overcome. Additionally, future research should explore the use of multimodal biomarkers, combining different types of measurements such as genetic, molecular, and imaging data. This integrated approach may provide a more comprehensive understanding of rare diseases and improve diagnostic accuracy. Furthermore, the integration of biomarker research into regulatory frameworks is essential for translating discoveries into clinical practice. Regulators need to develop guidelines and standards for biomarker validation and qualification to ensure their reliability and utility in clinical settings. References: 1. Chesler, N. 2018. Biomarkers in Rare Diseases. Rare Diseases and Orphan Drugs, 5-4, 161-162. 2. Gao, W. A., and Tift, C. J. 2011. The NIH Undiagnosed Diseases Program, Lessons Learned. JAMA, 305-18, 1904-1905. 3. Glicklick, R. E., and Dreyer, N. A. 2014. Registries for Evaluating Patient Outcomes, A User's Guide, 2nd ed.